Good morning, everyone. My name is Ita McGuigan. It has just gone 10 o'clock, so I'm going to wait for a couple of minutes while we allow people to join in automatically. It's a lovely, well, it's a coldish morning here. I'm in County Meath, and I know I'm delighted, actually. I'm just going to call out a few schools as you come in. Of course, I'm going to have to start with my home county of Monaghan. You might guess by this accent. So we're delighted to be joined by St. McCartan's in Monaghan. Also delighted to be joined with many schools actually from County Tipperary. You're very welcome, Ursuline Secondary School. A big shout out, of course, to Maynooth Post-Primary School, Carlow Institute of Further Education. We have schools in Laois. We have a few in Drogheda, Our Lady's College. We've Coláiste Doniska in Clare, County Tipperary. Kilkenny, welcome Loreto Secondary School, Mount Mellick, St Joseph's and Nina, High Tipperary again in Laois, Haywood Community School. We've schools in Roscommon and Strokestown, welcome Sculwera. Blakestown Community School, delighted that you can join us. You're just down the road. And Ballantyre Community School. We've St Mary's in Dundalk, County Louth. We're joined with, apologies, my pronunciation of Glenard, hopefully Glenard College in Arklow and County Wicklow, and many others. You're very welcome this morning. We're delighted that you can join us. My name is Ita. I work in the admissions office in Maynooth University, and I'm in the background. I'll be monitoring questions and answers. I know we have some schools who don't have the function to ask voice questions, but please use the Q&A question box on this Zoom, and I'm in the background answering questions. Delighted that Dr. Elisa Fada generously gave up her time, and I'm really can't wait to actually listen to this talk, because throughout the COVID pandemic, we have been listening to great information from leading Irish immunologists, including our own head of biology, Professor Paul Moyna, who you will have seen on or heard on News Talk and seen in prime time on the RTE News, on all over the media. But what I'm really interested to find out today is from the chemistry aspect. And with that note, I'm going to introduce Dr. Lisa Fada, and again thank you so much for your generosity of time to talk to our students today this morning. So Elisa, maybe before we start the presentation, maybe, I suppose students are thinking about their CEO options and choosing science on their CEO. Maybe tell us a wee bit about yourself and your journey to where you are now, and maybe some of the research that you're involved in. I'm sorry, I'm just throwing this at you. I didn't
yeah, that's fine. Actually, this is how I meant to start my talk. So first of all, I'd like to say I'm absolutely honored to speak to all of you today, and thank you for giving up your time to the school children that to listen to me today. Talk about my research and ongoing effort in understanding this virus, how this virus works, and how to curb its impact on our lives, which is already like enough. Like we already had enough of this. So I'm just like gonna probably start with to answer your question, Ita, with my first slide, which kind of like it's the same background that you have, which is the core of our university campus, our beloved library building, which is actually physically, or let's say geographically, at the core, at the center of the campus. So I'm to answer your question and to introduce a bit myself, I'm a chemist by training. So I have done chemistry in my undergraduate, but my research has also involved a lot of many aspects, and all these many aspects of many sciences have developed their contribution into my research over time through my PhD, through my postdocs, because I've done two, and through my work now. So my work is multidisciplinary, and that's what probably underlines the great utility of doing an omnibus in science, which encompasses different fields. So my work involves chemistry, biology, you mentioned Paul Moyna, with whom I collaborate and work with. So physics, computer science. So where do I, like, which are all like departments that are physically located, let's say on the North Campus, so on the right hand side of the library image now. So I'm based actually in what is called the Hamilton Institute, because my work is so multidisciplinary, but the common, let's say, denominator of all the people working in the Hamilton is to use computer to tackle different social and like scientific questions that of like main importance. So, and if you come over, and I invite you all to come over to visit our beautiful campus and to see what options you might get by studying at Maynooth University. For any query, please email Ita, like, which is like super knowledgeable about it. Don't email me about that unless you have questions specific to chemistry. So like, I would like to highlight one of the, the mascot of our campus, which is the famous library cat, who actually sits right before the library. Right? So you definitely will see him if you come over, especially if it's a lovely day like in the picture. Definitely is out there to get treats and be petted. It's very fuzzy. So so what I do actually, I use computing or large computers, and I'll show you what they look like through as a microscope to see what cannot be seen. And like this image that was actually shown on the cover of the New York Times 2 years ago now, like it's in October 2020, as it says here. It's from our work, like that we use computing to reconstruct and to image the coronavirus that is actually pestering our lives up to now. So in particular, what I do is I study sugars, and I explain to you what sugars have to do with the virus, like, which is a lot. And in particular, use the computers to study glycoscience. And glycoscience is a big word, and not a lot of people know it, like, or familiar with it. So don't feel like bad if you don't yourself, like lots of researchers don't even know, never heard of glycoscience. So it's the study of complex carbohydrates, and the word carbohydrates or sugars that actually sit or are linked or are connected through on the surface of other biomolecules that make up our cells and allow us to live, which are proteins and lipids, and now discovered last year, actually also RNA. So what, like to just like give you a bit of a brief about what I'm going to talk to you today. I'm going to talk about sugars, a lot of sugars, and why these sugars are literally everywhere. Is the most abundant biomolecule in nature, as you can see in the back of my background. There's there's grass, and that's sugars, like they make up the cell wall of plants. So also they're essential for viruses, especially viruses. They use any hosts, like in particular humans, hosts, like as we are concerned about it now. And why glycans are difficult to study. Like glycans are sugars, biological sugars is the name that we give to sugars when we talk about biology. And there can be considered as a black matter of biology. And what you need is most powerful, greatly powerful computers to see them, and normal, or let's say highly advanced microscope, such as, for example, cryo-EM cannot be used to see them. Computing viral glycoscience is what I do on a day-to-day life and what my lab is devoted to. And I'm gonna tell you today about our discovery and our colleagues' discoveries about the virus through computer microscopes. So like on the SARS-CoV-2, in particular COVID-19, which is the disease that SARS-CoV-2 causes. So I'm going to touch upon some vaccines and what are those vaccines that you all have, and some we're going to use in terms of drugs and diagnostics that stem from these discoveries, or can be also explained by these discoveries. So all of you know about the word carbohydrates. So some of you might have studied carbohydrates and know what the chemical structure of carbohydrates is. But like most of you, I'm sure if I could ask you, because there are so many, it's easy to interact directly with a class of 20, 30 people. However, 400 located in all over Ireland is a bit complicated. So I'm just like gonna ask you virtually a question, like you can answer it in your head, and if you have a question, like later, like you can ask me through Ita's question and answer session. So, but if you Google carbohydrates on Google, like what you get is actually, like, because I did out of curiosity, is 6 billion hits. I am sure that all of those, like 99.9% of those hits, are actually related to diet. So, and that's probably what you thought when I mentioned carbohydrates, like bread, potatoes, pasta. Some people might have thought fruits or like sugar, like just actually sugar that you add to your tea or coffee. So, and that's very true. Like carbohydrates are essential components of food. And in particular, like probably if you thought about bread, if you thought about plants like salad and whatnot, you thought about starch. Starch is one type of carbohydrate, like it's probably the most immediately clear connection to carbohydrate. And actually it's two types of carbohydrates, not one type, because everything is particularly difficult when we talk about carbohydrates, and let alone the chemical structure. So I just represented it here, the two types of carbohydrates that make up starch. So one is linear, so it's a polymer of different units. They're called monomers, okay? So they're repeated over 300 to 600 times, a very long chain of different monomers, that is amylose, and pretty much the same kind of arrangement, except that is not linear polymer, is branched, so it goes on to different directions. It's called amylopectin. So you definitely know, even if you couldn't recognize it right away, what is this? This is glucose, like, so is the sugar by definition. And why is this so important? Well, it's so important because glucose is the energy currency of our cells. So it's essential for our cells to work. And it's used by the cell to transformed by the cell through the very complex process that I'm not going to talk to you about, no worries. But for those of you who might be interested, it's called Krebs cycle, also known as the worst nightmare of biochemistry students, because it's so difficult to remember, requires a lot of chemistry knowledge. So, but through this process, the cell converts glucose into energy to live. So glucose, like for all we're concerned by now, is the energy currency of the cell, is the most important sugar for our lives. So, but you probably might be thinking now, there are other sugars. I've heard of other types of sugars, so other than glucose, and you'll be very perfectly correct. So let's think about like a common food, another common food, which is milk here. If I tell you milk, you probably and I ask you what kind of sugars there are in milk, some of you might immediately answer one sugar, that is lactose. So because of lactose intolerance, or because you just know about it, like there's a lot of people that are lactose intolerant, so they have like a bad reaction by drinking or eating lactose or ingesting lactose. So lactose is a disaccharide, means there has two sugars. One is glucose, our friend, and one is galactose, which is another sugar that is a dietary sugar that is used in our bodies. So like this might be getting already a bit confusing, because this, as you can see from the picture, from the chemical structure, is there shows like very high similarity between these two monosaccharides, like these two units. And this is very true, and it confuses most chemists themselves, not only people that do not do chemistry, and especially people that haven't done any chemistry. It's extremely difficult to draw or rationalize this structure. So don't worry about it. we have solved that problem. Now but the glycoscientist community has solved that problem, introducing a nomenclature or a way to represent sugars that is easier. So from now on, I'm going to represent sugars by color. So like glucose is a blue sphere and galactose is a yellow sphere. So there are other sugars than glucose, and that's a fact, and many more sugars. So if I use this symbol nomenclature, like this is a map of all the sugars that have been assigned as a symbol, which are not all the sugars that exist. New sugars are discovered virtually every year, and slowly but surely assigned like a different symbol. So you can see here, there's so many. We can get the glucose here, and you can see the galactose here, and so many others. So what are our bodies doing with all the sugars, and why do we make all these different sugars? Note that the type of sugars that we make is not the same type of sugar that other organisms make. So they're very species specific. So our cells, in fact, convert glucose into many different types of sugars, so reuse different dietary sugars in different ways. To what purpose? Well, to build very large structures that are complex, like you can see or use these little symbols and build them up as trees. Like these look like pretty much like, especially these ones, this type ones, they look like trees. And they're very flexible polymers. If you remember what I mentioned about starch being like a polymer, these are smaller than starch, but generally they're very, very flexible. So they move like say like a field and the plants in a meadow, like the one that are behind me. Like they're very, very flexible, and they're essential. They're a key to our life. So you might not recognize the names of this, but like if you look at those A, B, H antigen, these structures define or determine if you're blood group A, B, or O. So like those are sugars, all sugars that you probably didn't know yet. So they're essential to our lives, and they cover your cells like a fur. Like this is a cell from a bacterium that is used here because this bacteria have like very, very large sugars on the surface, so they can be imaged through a transition electron microscope here. So you can see here that this fur that looks like a fur to me, there are sugars, and there are sugars that can be represented through our like symbol nomenclature like these, and they are linked, so they are attached to proteins that are on the cell surface, through lipids or fats that are on the cell surface, and also, as discovered last year, through RNA that is also decorated with very complex trees, sugary trees. And this is an important process in biology because the cell itself, our cells, in particular mammalian cells, dedicate a whole lot of energy to build these sugary structures. And it's a very complex machinery that we're not going to go through it here, like in this case, but there's a lot of energy put into evolution to build these structures, which underlines the importance of glycans or biological sugars in our life and health and disease. So what are like these sugars do on the surface of the cell? So if we can represent, this is a graphical representation, obviously it's not like a cutout of the cell itself, but like it's an image that has been recreated. So of the cell surface, this is the membrane, this is all the lipids on the membrane. What are this fur? What is the fur do? What does the fur do? Like it just like is a way of the cell to communicate with everything that is surrounds it. So, and it allows it to interact with the environment and other cells and anything that needs to be communicated or with from the cell to the outside. And I mentioned here a friendly fur, or for a friendly furry coat, let's say. Why is that? Because it's built by our own cells. So the immune system actually does recognize like this sugars as a friend. So our immune system is not going to attack anything that has a friendly coat or that signals that that particular cell or protein is made by us. So it's not going to attack self. So it's a way of communicating to the immune system that everything is okay. So that's a way that a lot of, let's say, pathogens have exploited in order to attack us. So unfortunately, when there's a good side, there's always a bad side. So this friendly coat is actually used and attached to, or like, let's say, targeted by bacteria, by toxins, and by viruses. In particular, flu viruses, like, so how you used to get the flu, that's the masks, use of masks, appropriate use of masks throughout the world have lowered drastically flu contagion. But the flu virus actually, it gets you the flu, or gives you the flu through a handshake, literally, with those glycans on the cell surface. So what about coronavirus? Because we're not talking about flu here. It's equally sneaky, probably even sneakier. It doesn't use per se like a handshake to infect your cell, but it's even sneakier. So like, just to give you an idea, what is a coronavirus? First of all, this is an actual image of a coronavirus, very blurry and stained, well, stained, it is actually colored image of it. So it's an envelope virus, what is called an enveloped virus. So it's a family of, or a type of virus that has a genetic material in form of a single-stranded RNA enclosed in an envelope, or a spherical membrane, from which, like, proteins are called spikes, as you can see here in blue, stem out or stick out. And those are the proteins, or the like machinery that allows the virus to infect the cell. So, and they're called spikes. Well, because they look like spikes out of the envelope. So how did they get to us? Well, this is still not proven 100%, but like, let's say 99% chance that this is the way it went to us, as many other viruses that originate in animals, or like, let's say they're hosted by animals, or get to us. They come from bats that are actually magnificent animals. They are completely unaffected, and they just like have the virus and they don't care about it. The virus continues to live with them and replicate with them, so without affecting their lives one bit. However, sometimes these viruses jump to other animals. So in this case, researchers in this field have figured out that it probably went from a bat to a pangolin, which is an animal that is consumed in China. So it's a wild animal, and it can host coronaviruses. So, and why did I call it pangolin 0? Because it is the certainty of this path is related to finding a virus that is exactly the same as the human virus in pangolin. So pangolin 0 is most wanted. However, there's like viruses that have been found in bats and pangolins that are almost identical to our coronavirus. So that's very telling about the story of the pathway, how it got to us. So let's look at this again. Is a plastic representation of the virus that came out at the very, very beginning. Like, so it's not quite accurate, but it just renders, like, just to give you an idea. So this is the lipid membrane or envelope that encloses the genetic material of the virus. This is the spike proteins that come out, as you can see here in blue. These are in red. And why they're called spikes, actually, like I immediately thought about it in my walks, like in 2020, through the Phoenix Park in Dublin, which is pretty much what we were allowed to do back in the days of lockdowns. It's the similarity of this virus to chestnut tree seed case. Like you can see here that it has all the spikes. And it's quite similar, to be honest. I can give you a very clear idea why these proteins are called spikes, like they actually stem out. So, and where the sugars have any place in this? Well, coronaviruses use sugars to cover the spikes proteins. So the spikes that stem out of the envelope of the ball that you've seen are covered in sugars. The sugars are like, you can imagine it a bit like a candy floss, kind of covering this sticky protein that comes out of the virus. Like you can see here, different viruses spikes. So you can probably recognize HIV. So that's HIV as a spike protein, the HIV virus that gives AIDS. Then the H3N2 is a type of flu virus, like in particular is a type of protein that on the flu virus that is called hemagglutinin, and you see those sugars in green, and the SARS virus, which is our virus, and S stands for spike. And you can see here all the sugars, like in, again, a plasticky representation in purple. And why does it cover the spike? Well, do you remember that I told you that the immune system doesn't attack anything that has a friendly furry coat? Well, the viruses have evolved to hijack your cell to cover themselves in sugars for that reason, to evade your immune system, to cloak themselves with your own sugars or with the sugars of the animal that hosts it. So that if it's a virus in a bat, will be bat sugars. If it's a pangolin, virus in a pangolin, will be pangolin sugars. And hides itself from the immune system. So it gets into you through, like in our case, aerosol, and then gets into your body and starts replicating without your immune system knowing about it, unless you're vaccinated. So that's, of course, and we all are. So it's a cloak, and that sugar coat is a cloak, also called a shield, or technically a glycan shield. And you can imagine it as a, like a batty cloak like this. So it's important to study how this cloak is effective. So how well can this cloak hide the virus? The virus spikes, so which pretty much is the virus from the immune system. Well, that's extremely difficult because, as I told you before, those glycans, those sugars, biological sugars, are very flexible, like grass on a meadow. Like, so it's like trying to see them through actual microscopes that, and not optical microscopes, obviously, because of the size, but like different types of microscopes, such as I mentioned, cryo and electron microscopy, or different types of electron microscopy techniques. It's like a bit like taking a picture with a camera of a very fast moving object. So if the object in this case is a dog, runs faster than your shutter speed, what you get is a blurry image. Everybody amongst you have taken pictures, and I'm sure you've got loads of not well resolved pictures of people moving, or let's say your pet moving all of a sudden, and you have this blur. It's probably not to this extent, but this is pretty much what happens when you're trying to image glycans through any experiment. So how can we see them? How can we study the black matter of biology? Well, computing gives us a very good hand. So that's what we do at Maynooth University in my group. We compute glycoscience. We reconstruct these glycans, and we study them in real time, how they move, what structures, what 3D structures they have, and we make movies out of it that represent or allow us to understand their behavior. And in order for those physics fanatics amongst you, so like this is not actual very difficult physics. It's like Newtonian mechanics or classical mechanics, which I'm not going to get into details with it. But to give you an idea, it's a type of physics that people use to represent not only atoms in molecules, but also, which we do, but also study, let's say, planets' orbits, and or how to get, like, you know, like a probe to a particular planet, like as we've seen in Mars, the Mars rover. So it's a type of physics that allows us to study the very small to the very, very large, and is very effective at it in this particular case scenarios. So atoms are represented by beads, like in this case, like like hard spheres, bonds and angles by springs, and different type of approximations or potentials are used to represent electrostatic components and dispersion components. So like this for the very people that like to have a few more details on this. So this, based on this, we can actually reconstruct, and this is where physics enters my work, reconstruct a path or a trajectory and make movies out of it. So this is by solving the Newton equation of motion that is done by computer. Like we don't do it ourselves by hand. That would be incredibly taxing, well, impossible rather, and is numerical process that is solved by enormous computers. So what we need is, like, what we do is not possible on a laptop at all, but it's possible through the use of extremely large computers, like let's say like government-funded computer, like computing facilities that actually we sourced from Europe, like so that through funding through European ventures and through the Irish Centre for High-End Computing with ICHEC, which is Ireland has also a computing center of quite large size. So, and we also used for this particular, for reconstructing the shield or the say furry code of the COVID-19, of the SARS-CoV-2, we use the 8th largest computer in the world that is based in Texas. So what we did with all this, so like we actually took the structure of the spike, like so the rigid structure of the protein that was available, because it doesn't move that much, through one of those very powerful microscopes that is called a cryo electron microscopy. And this came out in February 2020, when everybody, all the scientists in the world were like jumping on this and trying to figure it out what to do and how to tackle this virus and to curb its impact on our lives. So, and we reconstructed the shield. So like when you see this, like when you see these two images, you realize immediately that the second one, the one from the protein alone, seems like shaved. Like it seems like part is gone, and you'll be right in thinking that. So the actual spike protein is not shaved, is actually covered in blue. Well, blue, we chose the color blue to highlight the presence of the sugars. But you can see now the candy floss, as my colleague Rami Amaro in University of California, San Diego likes to call it. It really looks like a candy floss, and it covers most of the protein with this friendly coat that hides it from the immune system. So I'll be back to that when we talk about vaccines. So we were able through this enormous computing power to reconstruct the virus in its actually more real form. So this is the fully atomistic system that you see the spikes with their sugars on it. You see the envelope and the spikes there. And this is how you can image like a virus, like a virus particle through computing, and this with the sugars on the spike. So this is a complete picture of what has been going around for the past 2 and some years now. So through computing, we were able also to see the dynamics of this spike, and this is something that is really interesting. So we used a different process or different computing platform or different microscope, computer microscope, if you will, for this, and we studied how the spike works. So as you can see from this movie, you can see that the spike that is actually turned around on an angle opens. And this the opening is essential because it allows the spike to promote infection. So allows the spike to attack or to the virus to infect your cell. It needs to open. So most of you probably have seen Alien, the movie Alien. Alien used to come from a pod. Think about it the same way. So the spike has to open and something comes out, which is not an alien, but in the case of the virus, but is a peptide, a protein part that sticks out and fuses the virus with your cell. So you know when the spike opens or the alien pod opens, trouble is in sight. So to do this work, actually, we used, as I said, a different computational approach, which is based on citizen scientists, and is approach that actually you can contribute to today. So to this effort. Here in this picture, you see all the people that have contributed to this work by lending their time, their computer time. For example, I have it on my laptop. Like you download what like an application called Folding at Home. And the Folding at Home will use the spare time that you're not using when you're not using your computer. So just like it would stop when you use your computer. So it's a bit like, you know, a screensaver or something like that, that runs the simulations or this experiments, this computational microscopy experiments, such as the one that I showed you earlier. So if you go on Folding at Home, if you're curious, you get like this big banner that's like, together we are powerful. And actually we are powerful together. So, and this is the contribution from computers all over the world, and loads in Ireland as well, have created the most powerful supercomputer on the planet, which is exascale computing. It is by far the most powerful supercomputer on the planet, which for those computer scientists, like, or budding computer scientists, is like called, this platform is called distributed computing. So you can use your PC to help fight COVID-19. You can get all the information there if you're curious. So I encourage you to check it out. So going back to how can HPC simulation help? So what kind of work, like what kind of questions have we answered and we're still trying to answer? So first question, as I said, very early on in our research, starting in like, you know, February 2020, how good are the sugar or this coat, this glycan shield at hiding the spike protein? Well, actually the answer is very good. So we found that more than 62% of the surface of the spike is covered by this candy floss. So, and this is very good for the virus, as you can imagine, because its coat is quite effective as hiding it. So it gets through your immune system undetected. But if you can see here, like, you know, and you remember that the protein or the spike protein is actually represented in cyan, you can still see some cyan bits. And in particular, this bit that is called a receptor binding domain. And it's one of those flaps of the alien pod that opens. And in this case, it is open. So when it is open, it actually sticks out, as you can see, and it's very visible. And it can be seen by the immune system as foreign. So the immune system sees this and he said, well, this is not human. What is this? And it just recognizes and attacks it. So there is a recognition that goes on by antibodies, and it can be exploited for vaccine design. And this is exactly what has been done. So the spike protein or the RBD alone, or this receptor binding domain, just a flap bit, is a great construct for a COVID-19 vaccine. This is why it works. And this is why all the vaccines that we had or are approved in Europe are based on the spike protein. So I took this screenshot from my booster vaccine that actually is Moderna. And that's why it's called spike-vax, because it is a spike protein that is not in a virus, is just a protein, and allows your immune system to recognize that part, those parts that are in blue or light blue or cyan. And when the virus gets into you, is already recognized, and your immune system is already alerted and ready to fight. So that's why vaccines are so effective. Most of these, like all of these, actually contain the spike protein. One of these is only a receptor binds the domain, is the vaccine that is designed and produced by Cuba. It's Soberana and Abdallah. So like the spike protein, or the understanding how the spike protein works, pretty much set up a way to design these vaccines. So, and that's why they work. So the question too would be, how are sugars or the glycan shield functional? So what does this mean? So like, are this furry coat just sitting there, just protecting the virus from the immune system, or regulating its recognition by antibodies or immune response, or actually is integral part of the machinery? So it allows the flap to open, or and the alien to come out of it. Well, it does. This is like, we discovered this through enormous amount of computing, and this is the only time, like, or the first time this was recognized for a virus. Like, so this is that the shield, or the sugars, are integral part of the mechanism of working, like how the spike works. Like without sugars, the spike doesn't work. So this is like how do they work? Like if you see like this, the sugars here are represented by glossy like thingy, and you can see here in particular the purple sugar here props up one of the flaps. That is, like in this case, the spike has only one flap open, like of the pod, and he actually helps it up in an up position. And this allows it to stabilize the open form of the spike, and to make it very, very infectious. So you can imagine if you, yeah, Ita?
Yes, sorry, Elisa, I was going to wait until you finish that slide. We have a couple of questions in, and I was wondering, would it be...
I can stop.
Yeah, oh, brilliant. Thanks a million to St. Joseph's CBS. You're very active this morning. I have two questions, and if you're comfortable with answering them now, that'd be great.
Sure, yeah.
So the first one is, could you clarify how your research could help with future pandemics?
Yeah, like it's just like, it's, so when I get to the variant, probably, like, so like it would be more clear, like it would be that okay? Like,
perfect, yes. And the other one, possibly you may be able to answer now. I could take a stab on it, but my scientific knowledge just mightn't be up to yours. Of course it's not. Is there any way to remove the glycan from the coronavirus?
Next slide.
Perfect. Okay, thanks a million, St. Joseph CBS, and encourage everyone else, if you wish, to put the questions in there.
Those are extremely good questions, and I'm delighted that people are actually following so closely, because they're very pertinent questions that we ask ourselves all the time.
Others, please feel to drop in questions, and I'll monitor them.
Please,
and I'll
yeah. So if there's anything you don't understand, I'll just stop me right away, and I'll explain it again. So based on that question, there's a lesson learned too, is that by removing or changing the type of sugars, you can actually weaken the spike and make it useless, pretty much. To some degree, you can make it useless, reducing infectivity. So there are some approved drugs for other diseases that do this job. So they act on the size of this glycans, or the sugars, so that you would change the type of sugar sugars in the glycan shield that weakens the spike. And this is a work published in the Lancet, like in 2021, that proves that it actually changing this using these drugs is effective in reducing infectivity, and it's pretty much somehow well, you cannot like you cannot literally shave them off, but like that's not possible. But it is possible to trim them or to leave them very immature in a way that actually are bigger, but they don't work at all. So this is a very, very good strategy, and it's been explored. Like Lancet is one of the top, if not the top, medical journal in the world. So lots of people are onto it now. So this hopefully answered the very pertinent question about changing the glycan shield or removing the glycans. So going to the other question, the variants. So this is, you see, like future pandemics, it's something we live every day, literally, and gives us headaches or dread to open the news, like mostly dread. And it's like yet another variant. So what are the variants? Variants are mutants. So that occur and actually, like they say, take or substitute a virus that is not as good. So what makes a virus good? A virus that replicates faster, so that is like more effective at infecting, more effective at replicating. This is like a virus doesn't want to kill you, because killing the host is not generally like a best strategy to a replication, but it just like it wants to replicate. That's its goal in its well, if you want to call it life cycle, but it's a mandate. So how does it do that? Well, in order to replicate faster, it needs to get onto the cell more effectively and attach to the cell more effectively. So that's what greater binding to the host cell receptor means, like getting a spike that attaches to the host cell more effectively. So, and it also, one other aspect is a better hiding from the host immune system. The more it hides, the tougher is for us to get to train our immune system in order to see it. And that's the problem when mutants occur, because they might change actually the structure of the proteins. That's what a mutant is. So if you think, like, if you look at this is actually the code, the protein code that gives you the structure of the spike protein. And you see all these letters represent an amino acid. So this blue are where those glycans or the sugars are. So during replication, you can have mutants. So like some of these letters can be changed, and this has different effects. So in this map, you can see that it's taken from this paper. You can say that some mutation, let's say a D to G, or or N to Q, might increase infectivity or decrease infectivity, and or change like the interaction with the immune system. So like it would change kind of the virus impact. Might change, might not change it to the good or the bad. So obviously, when it changes to the good, that is the virus that suppresses all the other viruses, especially when it changes to the good, to the very good. And this is what happened to Omicron. This is Omicron. So like this is a spike, Omicron spike. So like this is now the virus that is everywhere. And why is it so effective? Because you can see all these blue little bits are like the mutations. So the amino acid or the letters that have been changed relative to the original virus that we had at the beginning of the pandemic. So you can see that a lot of mutations are here actually on the flap, on one of the flaps. Why? Well, because it binds better to the host cell. And that's why it's suppressed. Everything else is very contagious. It binds super fast. So it's suppressed. All the other viruses, the cousins viruses that were around, or the brothers, like if you want to, like, because they're very close in the family. So, we get why like some of the variants have a greater binding to the host cell receptor, but how about the hiding from the host immune system? Well, this is a business of the glycans. And we looked actually through the whole family tree. So if you think about the ancestral tree of the virus, of the SARS-CoV-2 virus, and we just like took reconstructed where the glycans, where the sugars were, this is all the changes that along this family tree we have seen. And actually, there's a very recent change that our virus, including Omicron, has relative to an older version, an older cousin, not directly related, but incredibly similar, that is SARS, that had came out in 2003 with contagious pockets in China and in Canada, where I used to live at the time. And I remember, so I lived through 2 of these SARS things, unfortunately. So, and this is due to a mutation, like of one of the letters that went from T to A. So the change in the shield, like, so changing the coat, like say, you can think about like a flap going back, like if you think about it, like glycoshield is a coat, change a bit of the model of the coat. It's actually unique to the virus that we have in humans, like, and is not present in the viruses that have been found so far in the bats and in the pangolin, and not in the old SARS. So the point is like these changes are quite complex and not only allow the virus to hide differently from the immune system, but also to function differently. Because if you remember, I told you that the sugars are actually essential for working. So in particular, it allowed the glycan that disappeared is a yellow one. It's gone. And it left kind of like a pocket, like if you imagine this pocket without the glycan that is free now, that is not occupied. And this particular loss of this sugar is actually very useful, because as you can see here, probably it just kind of flaps over one of those or just like covers one of those flaps, and it just prevents it from opening, like if you remember the pod before, making like this loss advantageous for replication. So this is actually a bad news for in our case for the SARS-CoV-2 that acquired this this feature in particular. This change might have resulted of a higher infectivity of the SARS-CoV-2 relative to the SARS-CoV in 2003. So, but also allowed it to bind to the fur of the host cell. So that cleft can be actually occupied by different glycans or sugars that are on your cell, making it binding more to the host cell, which is also bad news. And we've seen through this our work that it can bind blood group antigen A. So like in this case, like explaining why some people that have different blood groups might be more, let's say, like infected than others. Like this is something that was at the beginning discussed. And some glycolipids or fatty sugars that are on your cell fur. And this is actually a very important also features for tackling through drugs, because drugs can be designed when you have the structure of the cleft like we do, and see how and what it binds to design a drug that prevents this by occupying that cleft. So this is a lesson learned number 3 through computing that allows us to progress in different therapeutic strategies. So, and also based on this, and this is my last bit, like our colleague and friends like in the University of California, San Diego, Rami Amaro's group, based on this work, they actually developed a diagnostic. So you have seen all lateral flows, like, so this is a bit of a different lateral flows. It's all like fast, rapid testing based on sugars. So like you see, this is part of the the furry coat of the cell, and they transferred all the sugars from the furry coat to a slide, like that is on your lateral flow testing device, that are selective to bind the spikes on the virus, and it can be actually used through antibodies for a negative and positive. Positive if you have SARS-CoV-2, negative if you have none or another virus that is not SARS-CoV-2, because these sugars bind SARS-CoV-2 exclusively. So to conclude, how computing, or the computing microscope has been useful for our research or insight, getting us insight into COVID-19 research. So we found that the shield, or the glycans, or the sugary coat is essential not only for immune escape, but also for the activity and infectivity of the virus. And I hope I explained to you why the spike is a very successful construct for the vaccines that are so effective now. So the lesson 2 is like the type of sugars, and actually the shield itself, or like its nature, like its density, like how much is covered, how much is not covered, can regulate infection. And like, let's say, modifying this coat itself by reducing its size or increasing its size to immature glycan, what is called. That's very technical, but allow me that, like, it's leading to a therapeutic strategy that prevents the infection in the first place. So, and also this particular, like I showed you, that this particular virus has the ability of latch on to the cell, the cell glycans, and this increasing its infectivity. And this can be prevented by designing drugs that are built to bind or to swap like places with this, like a pink sugar, and prevents the pink sugar to bind and for the virus or the virus to latch on to the cell. So with that, I'd like to thank all of you for your attention. I probably took loads of your time. Just went on like talking a bit too much, which is my problem, like one of my, let's say, like characteristic. I can talk a lot. So, and all the computing resources that we had, and Ita for organizing this. It's been a pleasure for me to talk to you. And if you have any question, I'll be happy to take them.
Oh, that was amazing, Lisa. I learned so much. I found such an insight into that. And what I really, really loved was, your growth from chemistry, biology, physics, computer science, the whole STEM field in the, I suppose, investigation of COVID, and hopefully the solution, if there ever is a solution, which brings me on to, and apologies if I've missed this, St. Joseph's. I have to say, your top school this morning. Another question in,
Sure.
And do you anticipate any more variants, Lisa?
Oh, geez, that's a very difficult question. So I am I'm not a virologist, so that's missing in my list. So or worse, an epidemiologist. Like you probably, if you follow the news, everybody kind of it's a new epidemiologist or a new virologist. Everybody that is kind of claims knowledge about these things. I don't claim any knowledge about these things. It's natural to think that the more the virus circulates, the more the chances new variants are going to come up, as this came up. So I would say that until the world is vaccinated, we'll be plagued with this. So I cannot encourage anybody more to consider, for example, UNICEF Ireland effort in donating vaccines to third world countries and promoting that until third world countries or or governments are able to subsidize either vaccines or facilities to make vaccines in third world countries, because until we are all vaccinated, this will just continue to come around and come up with new strategies to get to us. Because viruses are not good or bad, they're not living, they just do a job. And their job is to replicate. So there are no more, there's no ethics in this. It's just that they will continue until they can.
Yeah, and just on that, I know schools, and in particular, I know that I was in Maynooth Post-Primary School before Christmas, and students there are doing excellent work on trying to get the vaccines to those countries who aren't as fortunate as us here in Ireland. And so well done to Maynooth Post-Primary School, and indeed other schools around the
Excellent,
country.
amazing. Like, you know, it's something that, you know, like I'm telling to just like, you know, raise awareness that this is should be everybody's, especially government, first concern together with their own population, because their own population will be continuously plagued with this until we vaccinate the world. And it's nobody's fault. Like, as I said, there's no ethics. Like the ethics doesn't enter the realm of virology. A virus does its job and it will continue to do it successfully. That's why they're all the organisms that always lived. You know, we don't know if it's the first organism, that's a big word, like, but it's the first, or the, you know, like it's just like super prehistoric. Like, but again, I'm not an evolutionary biologist, so I don't know, like take my words like with a grain of salt, but it's extremely efficient at doing what it does. So, well, as you all have seen.
I would like to take this opportunity on behalf of all the schools, and hopefully they all enjoyed it, just to let the schools know that I will send this recording out, and thank you for Elisa for allowing me to record it. All schools will have access to this. And just to let you know, schools, I will also send you a link to another talk we're having by a colleague of Elisa's in the Maths and Stats department, Dr. Niamh Cahill. You may be interested in that. I'll just, when I'm sending you the recording, I'll put a wee paragraph about it. And if it's of interest to you and your students, please feel free to sign up. On behalf of everyone attending here today and the admissions office, Elisa, I just want to say it was a pleasure working with you on this. I hadn't to do much, to be honest, but I really want to thank you for your insight. I certainly learned a lot, and it was brilliant listening to you. So I just want to thank you for your time.
Well, thank you
Today.
Thanks so much. It was a privilege, like, to speak to all of you. So I really truly hope to see all of you or some of you in Maynooth, like in the coming years.
Yeah, listen, best luck everyone. Teachers, go and get a hot cup of tea or a coffee, and thank you so much. You've got my email and details if should you need any information on STEM at Maynooth University. Thanks everyone. Thank you, Elisa.
Thanks a million. Bye bye.